I believe it is important to classify which lipid that we will be discussing when talking about the lipid bilayer. Lipids are water insoluble, which means they do not dissolve in water. For this reason it makes a great cell membrane. As you can see from the picture here, there are three main types of lipids, but the type of lipid that we are going to talk about is the middle one, the phospholipid. The lipid bilayer is made up of phospholipids. They have two parts to them. They have a head where in the picture here you can see it as a blue singular circle. The second part is their tail where in the picture is depicted as two curvy dark green lines. The heads of the phospholipids are hydrophilic and the tails are hydrophobic. This makes the phospholipids amphipathic, which means it has both hydrophilic and hydrophobic components. Hydrophilic means water-loving, so the heads will be facing the aqueous environments. Hydrophobic means water-fearing, so the tails will be pointing away from the water. Phospholipids are bulky. They have one hydrophilic head and two hydrophobic tail. Here in the picture you can see the head of the phospholipid is labeled and the two tails are as well. One of the tails usually will have a double bond present and it creates a kin. You can see that the right tail has a little kink towards the bottom and the other tail does not. The double bond means that it is an unsaturated fatty acid. Since the tails are hydrophobic and they want to get away from water, they will pack together with their tails facing each other. The heads will be out towards the environment or inside the cell. Here we have a picture of a lipid bilayer. A lipid bilayer is made up of two rows of phospholipids, which you can see here where the pink are the phospholipid heads and the blue are the lipids tails. It will have a hydrophobic inner part which includes the tails of both rows. It will also have hydrophilic outer parts which has the heads of the phospholipids point to either put into the environment or inside the cell. This information will create a hollow-like ball called liposome. Lipid bilayer generally are flexible, which means they can bend and distort easily. It is also important to note that both the lipid bilayer are different. This means the outer part is different from the inner layer. The lipid bilayer creates a permeabiliar membrane. Micelle occurs when detergents are added to the cell. When they are added they cause the phospholipids to freak out. Since the tails are hydrophobic, they will do everything in their power to get away from the water. In this picture you can see a detergent was added. The phospholipid heads are silver and the tails are blue. The tails got away from the water and are pointing towards each other to form a barrier and protect themselves. This causes the phospholipids to form a circular shape. The concept of a liposome is that the lipid bilayer rolls into a ball to have no free edges. Not having any free edges means the bilayer is self-sealing. When the bilayer rolls into this ball it conserves energy. This is a good thing because then the energy the cell does have can be put to better use. Here in the picture you can see the heads of the phospholipids are touching and creating a circular shape. Membrane lipids are phospholipids and glycolipids. These do not do well in micelle because the double bonds found in the tails make it too bulky in the center of it. I like this picture I found because it shows you the phospholipids and glycolipids broken down, and it goes into great detail on what each of them are. Membrane components are sterol cholesterol and glycolipids sugars. These sugars that are in the cell membrane are complex molecules that are made from multiple individual sugars that are linked together. These complexes are called oligosaccharides. There also are glycoproteins as well found on the membrane. They have larger and more complex sugars that are attached to their exterior face. These form a sugar layer on the outer part of the cell and that is called a carbohydrate layer. This layer will help protect the cell from mechanical or chemical stress from the environment. This layer is most useful for cells that move. There is a specialized protein called lectin that will recognize and bind to individual sugars on the lipids and proteins that are a part of the carbohydrate layer. This picture is similar to the last slides because it showed you the different glycolipids and sterols that are found in the membrane. It's important to talk about the membrane as a whole. The components are hydrophobic, so this means they will never just pop off and float away from the membrane. They also will shield themselves by huddling around other hydrophobic things in the membranes too. They want to be away from the aqueous environment and be safe in the cell membrane. The membrane is flexible. This means it can bend and distort easily as discussed on a previous slide. Here this is a picture of the phospholipids, all the membrane proteins, and other membrane components together to get a better pictures on how they fit into the membrane. Lateral diffusion is when the membrane components can move in the membrane forwards, backwards, and even side to side. In this picture here you can see here the dark pink head on the phospholipid that is on the left side of the arrow and it starts off as the second lipid in. Then when lateral diffusion occurs, it switched places the lipid next to it because now it is the 3 lipid in lateral diffusion. The membrane components will never flip upside down. That is called something different, which will be discussed on the next slide. Lateral diffusion is the reason that the membrane is 2D fluid. Even though it is not easy to do, sometimes the membrane components can flip upside down. When this occurs, this is called transverse diffusion. Here we have the similar situation as lateral diffusion, where the bright pink head on the left of the arrow and it starts off in the second place. Then when transverse diffusion does happen, the lipid switch when the lipid that is across from it rather than next to it, like lateral diffusion. This takes a long time to occur. When it does occur, an enzyme called flip passes is needed to fix it. Flip passes corrects the components to be in the correct phase. When talking about fluidity in cell membranes, it refers to how loose the membrane lipids, proteins, etc. are packed together. More fluid in the membrane means that the cell is less dense and will not hold its shape as well, and it also means that the membrane is easier to float in. It is important to note that different fluid items are needed for different environments. Cells can regulate their membrane fluidity if the environment or function happens to change. In this diagram here you see a lot of space between the phospholipids. The purple are the lipids heads and the black curvy lines are the 2 tails. The blue shapes are membrane proteins. When there is more fluid in the membrane, it will looks like this, where the lipids are not tightly packed together, therefore allowing them to move more easily and not be able to hold the proper shape it needs to. The membrane also will not be keep the water away from the hydrophobic tails like it needs to. When the membrane is less fluid, that means that the membrane lipids are closer together and tightly packed, making it denser. There are a number of different factors that play in making a membrane denser. You have the length of the tail and the number of double bonds that play a role in how tightly packed the membrane can be. Less kinks would make the membrane less fluid than a membrane that had several more kinks in the lipid tails. If the lipids are short, then they will also not be able to pack tightly together. They have to be long. Less fluid membranes also have a tough time in cell division. In this diagram here, you can see there is no spaces between the phospholipids. They are tightly packed together, which means nothing is going to get into the cell easily. Bacteria can regulate their fluidity. They can change the double bonded carbons in the hydrophobic tails to either make the membrane more or less fluid. They also can change the tail length as well, depending on whichever direction they are trying to go with fluidity. Animal cells regulate fluidity differently than bacterial cells. We cannot change the length of the lipid tails or the double bonded carbons. We regulate fluidity using cholesterol. Typically, the cell membrane in humans is about 20% cholesterol. However, human cell membranes do differ on fluidity, so this percentage could change. Cholesterol stiffens flexible tissues, so a lot of cholesterol is not good for the body because it will harden the arteries. More cholesterol makes the cell membrane more dense and stiffer. Less cholesterol in the membrane makes it looser. Cholesterol is found in the gaps that the double bonds on the tails create. In this diagram you can see here the cholesterol is a brown color. You can see it placed in between the phospholipids tails where the kinks are. This here will help regulate the fluidity of the membranes. The next membrane component I would like to discuss are membrane proteins. Membrane proteins make up approximately 50% of the membrane. The membrane proteins' main function is to transport. They transport things like ions, sugars, metabolites, etc. that cannot cross the membrane by themselves. Membrane proteins also act like anchors, receptors, and even enzymes. Membrane components are cell-specific, so it all depends of the function of that particular cell what you would find in the membrane. You will also see amino acid chains just embedded into the bilayer too. So here in this picture you have all the membrane proteins I mentioned. You have the transporter which is green. You have the receptor that is red. You have enzyme that is blue. And you have the anchors that are purple. Transmembrane proteins are embedded into the interior part of the bilayer where the hydrophobic components are located. They will extend through the entire membrane and the two end regions of them are hydrophilic. In this picture you can see the transmembrane proteins are a little blue color, and you can see they are embedded into the membrane because you can see a layer of phospholipids in front of it. Two more membrane proteins are integral and peripheral. Integral proteins are not covalently bonded to anything on the membrane. They are however embedded into the membrane and can only be freed from the membrane with the use of detergents. Peripheral proteins are only covalently bonded to a phospholipid's head. It does not go through the whole membrane like the integral protein. In the picture you can see the integral protein has an opening like a channel. The peripheral protein is embedded into the membrane and just in the heads part of the phospholipids. Before we move on to the next topic of clathrin-coated vesicles, we must first talk about coated vesicles. When a vesicle is coated, it is typically coated with a protein coat known as clathrin. This protein will form a cage-like structure around the vesicle. This helps with a number of different things such as form buds, hold the vesicle's shape, and make sure the vesicle is carrying the correct cargo it is supposed to be carrying. In this diagram here, you see the protein coat that like red things that are sticking out of the vesicle's membrane that is yellow. This down look a bit like a cage around the vesicle here. You see that it is helping hold its circular shape. Now we will move on to clathrin-coated vesicles and its formation, docking, and fusion processes. Clathrin coats vesicles are found in two areas in the cell. It can be found budding off of the Golgi for secretion and it can be found budding off of the cell membrane for endocytosis. We are going to focus on the formation of the clathrin-coated vesicles budding off from the cell membrane. When the clathrin-coated vesicles is starting to form, it starts off as clathrin coats pits. A protein called dynamin then comes in and will constrict the neck of the vesicle by forming a ring around it. This will cause the vesicle to break off from the cell membrane. Another protein called adaptin is needed for the formation of the vesicle. It helps secure the clathrin cage to the vesicle and it helps choose what the vesicle should be carrying. Adaptin will trap the cargo below the neck of the vesicle. Finally, there are transport signals that help the vesicle know what the target it is meant to go to. Usually when it's a different target, the transport signal is binded to the cargo receptors. In this diagram you can see the different stages of the vesicle formation. First you see the adaptin is a light green and it's covered by the clathrin coat that is a dark green. The cargo receptors are a blue color and those are attached to both the cargo that's a red color and the adaptin. In the next stage you see dynamin is yellow and it's wrapped around the neck of the vesicle helping it bud off from the membrane to go to its targeted destination. The next step show the clathrin coats vesicle completed with all of its parts intact. The next stage is the I-coating process for docking and fusion, which we will talk about on the next slide. After formation, the next step is docking. In order for docking to be able to occur, the target organelle must recognize the membrane of the clathrin-coated vesicle. Rad proteins are located on the surface of the vesicle that act as a clue so they can be recognized by tethering proteins. There are different types of rad proteins, so each one is like a membrane identifier. The tethering protein will bind into the rad proteins. Snare proteins then will bind to the vesicle's membrane and the target's membrane. This is like a second anchor like the tethering proteins are. In this diagram, this is cool because it shows the whole docking process broken down. You have the snare proteins depicted in the purple L shape that is attached to the transport vesicle. Then the vesicle is attached to the tethering proteins that's attached to the target membrane. You also have the rad protein that is attached to the tethering protein and the target membrane too. You then see in the step before fusion, the tethering protein is gone but the snare protein is still there anchoring the vesicle down to the target membrane. The final step is fusion. Fusion can only happen once the vesicle is fully docked and anchored down on the target's membrane. The vesicles will then shed their protein coat or cage structure. Before the fusion process can actively start, a cellular signal must be given to the vesicle. Snare proteins then will twist and winch the vertical closer to the target's membrane. When it is close enough, the vesicle will then fuse with the target membrane and it becomes part of the target's membrane now. Then the cargo that was in the vesicle is then released into the target's membrane. In this diagram here you see the vesicle has shedded its clathrin coat. The snare proteins are green and purple and they are pulling the vesicle close to the target membrane. Then finally you see fusion happen where an opening in the vesicle appears making it part of the target's membrane now. The next topic we are going to be discussing is the formation of the three different protein filaments that are in the cytoskeleton. Intermediate filaments are made up of various fibrous proteins. Microtubules are made up of tubulins, and microfilaments are made up of actin and are also known as actin filaments. This is a picture of the three filaments side by side to show their differences. We will go into more depth on their structures and formation in the next several slides. Out of the three filaments, the first one we will be discussing is the formation of intermediate filaments. These are part of the cytoskeleton fibers. Individual fibers will begin twisting together. You can see this in the picture on the fiber that is labeled a monomer. When two fibers twist together, proteins will come in and wrap around them, forming a dimer. In the photo, a dimer looks like a braid. You can see the two individual ones wrapping around each other tightly. Then two dimers will come together to form a tetramers. Here you see how more dense tetramers are than the dimers are alone. Finally, the tetramers will come together to form a rope-like structure. This is shown as a filament in the picture, and it is at the very bottom of this picture. You can see through each step of its formation. It gets denser and denser. Next, we will discuss the structure of a IFS. Its function is to help the cell withstand mechanical stress. Without IFS, the cell could tear. Examples of mechanical stress would be stretching, pulling, and squeezing. Where you find the most IFS are in neuron axons, muscle cells, and skin cells. Out of the three filaments, IFS are the toughest and most durable. When in the cell, IFS surround the nucleus, then extend outwards to the cell's membrane. They will then anchor onto either side of it. IFS are also found inside the nucleus, a part of the nuclear cortex. They are in the main component called nuclear lamina. There it helps strengthen the nuclear envelope. The head of IFS are globular and can interact with many other things. Some example of things they can interact with are other IFS, anchoring points, and organelles. This is a picture here of IFS in epithelial cells. On the left side you see the blue curvy lines that are all over. That is IFS. You can see mechanical stress being put on the cells, but IFS are helps them not break and withstand it. On the right are cells that do not have any IFS present. When mechanical stress is put onto these cells, they just popping. They could not handle the stress alone. Plankton is an example of an accessory protein that can be found to help strengthen IFS. It helps strengthen and cross-link other IFS together. This will make them stronger. This will also strengthen the cytoskeleton against mechanical stress. In this picture here you can see plankton is a light blue color. You see it is attached to IFS that are a darker blue in the top right-hand corner. There are four main classes of IFS that are important to mention. The first one is keratin. Keratin is found in epithelial cells and it helps in the distribution of stress from stretching on the skin and the gut. Next is vimentin, and this is found in connective tissue, muscle cells, and cells that support the nervous system. Next are neurofilaments that are there for structural support to neurons. And lastly, nuclear lamins, and as discussed in the previous slide, this strengthens the nuclear envelope. Out of the four classes, 3 are cytoplasmic and there is only one that is nuclear. I am sure from this title of this picture you can guess that it is lamins. Lamins must be able to disassemble and reassemble every time cell division occurs. For lamins to be able to disassemble, phosphorylation must occur to weaken the affinity of them. For the lamins to be able to reassemble, they must dephosphorylate. In this picture here, you can see this green woven sheet of lamins. Once it is phosphorylated, you see the individual strands better and see that it clearly disassembled itself. If the process was reversed, it would be put back together and tightly together again like it started. We move on to the next filament in the cytoskeleton, and that is microtubules. We will first talk about the formation of microtubules. First, two tubulin proteins will come together to form a dimer. Dimers will have an alpha and beta end. The dimers will then come together to form a protofilament. These interact through noncovalent forces to do so. When 13 protofilaments come together, they form a single MT. In the picture here you see the beta tubulins are blue and the alpha tubulins are red. Then when they come together to form protofilaments, you can see the alpha and beta tubulins alternating. This is so then when they go to form a single, hollow MT, one end with me all alphas and the other will be all betas. Next, we will discuss the structure of MTs. When the 13 protofilaments come together to form the MTs, they create a hollow tube. They will have a beginning of only alpha subunits and an end of only beta subunits. Alpha subunits will have a negative charge while the beta subunits will have a positive charge. These two charges give the MT its polarity, therefore, giving MTs their function. Their function is to create lanes for cellular transportation. The polarity helps define the direction for transportation on MTs. In the picture here you see the alphas are light blue and the beta are dark blue. This picture shows you a side view to show you that MTs are hollow. Centrosomes are where the initial MT tube forms. Tubulins cannot just form new tubes out of nowhere. Centrosomes have lots of ring-shaped structures in them and they are where the nucleation site is. The nucleation site is where the formation of MT starts. The negative end will be embedded into the centrosome and the positive end will be going out towards the cell. In this picture, the centrosome is the big green sphere. Coming out of the centrosome that is a darker green and looks almost like spikes are the MTs. In the centrosome that doesn't have any MTs coming out of it, you can see the nucleation sites and they are little red circles that are all over the centrosome. Dynamic instability is important to know when talking about the structure and formation of MTs. Dynamic instability in this context means that MTs can and will disassemble randomly. This means that dimers will fall off either partially disassembling the MT or they will fall all the way off completely to the centrosome. GTP helps back the dimers tightly together, while GDP causes them to break off. Polymerization is when dimers are added at a rapid speed and a GTP-CAP is formed. Depolymerization is when the GTP get hydrolyzed and the ends of the MTs start to fray. This causes the MT to disassembled and once it starts that process it is hard to stop it. To stop the MTs from falling apart, they have to be anchored to something. In this picture here you can see GTP is binded alpha tubulins that is a dark blue. You can see that polymerization helps add dimers and make the MT longer. When depolymerization occurs, the MT falls apart and will have GDP present. GTP like polymerization does. Motor proteins can be found walking along MTs and contribute to intracellular transportation. The motor proteins will be carrying the cargo to their destination while walking along MTs. Kinesin and dynein are two motor proteins that can be found walking along MTs. Both of these proteins are dimers that bind to ADP and cleave it for energy to move. The heads of them will bind to the MT and the tails of the motor proteins will bind to the cargo they will be carrying. Kinesins will travel to the positive end, while dynein will travel to the negative end. I like this photo because it breaks down the stages of the motor proteins walking on the MT. You can see kinesin is the color green. You can see the head of it is two little green circles and the tail extends off from it and is carrying some cargo. Dynein is the same but in the color red. The cycle for motor proteins to move along MTs or any filaments is pretty straightforward. Find the motor proteins will bind to ADP. Then they will cleave the ADP to get energy. They will release the now ADP that will result in a conformational change to the motor protein. This conformational change allows the head of the motor protein to let go of the MT and take a step forwards. Once it takes a step forwards it will then rebind to the MT and the cycle will start all over again as it makes its way down the MT. This picture narrows down more on how the motor proteins move when using ADP. Here we have one protein on the top of the MT and the other one on the bottom. They both are binded to a vehicle that is carrying some sort of cargo. The motor proteins will bind to ADP and cleave it down to be ADP, causing one of the domain heads to move. The ER and the Golgi both are positioned in the cell by MTs. The motor protein kinesin will attach to the face of the ER while the motor protein dynein will attach to the Golgi. If something does happen where the MTs disassemble, this would cause the organelles to change their placement in the cell. But once the MTs reassemble, they can fix them and reposition them again. In this picture, you can see kinesin is the color red and dynein is purple. You can see they both are starting from the same place but are moving away to their different organelles. The final filament in the cytoskeleton we will be discussing is the actin filament. A single actin filament twists into itself creating a globular subunit. Then it will bind to ADP. Every free end of actin will bind to ADP. Then the ADP-bounded actin will bind to another ADP-bounded actin to create a dimer. Dimers will then come together to form a filament. These filaments will have a positive and negative end like MTs do. In this picture you can see the ADP-bounded actin have a purple center, while the actin that doesn't have ADP bounded to them are just a solid red on the right side. Moving to the left you see the ADP-bounded actin will come together and form dimer. These dimers then come together to form actin filament. Next we talk about the structure of the actin filaments. Actin filaments are thin and are compared to be thread-like. They are short and flexible. There is an abundance of actin in the cell. Polymerization is when the actin binds to ADP. This causes the actin to become more tightly compacted together to form a filament. Depolymerization is when the ADP is cleaved and ADP is released from the actin caused it to disassemble. Since they do not have proteins to help stabilize them, they can disassemble easily. Most of the actin that is found in the cell exists as free monomers, which brings us to our next topic. This picture is just a simpler version of the last one on polymerization and depolymerization. Here you see polymerization holds the actin together to form filaments and depolymerization breaks the actin filament apart creating free monomers. The amount of actins that exists as free monomers is high. There are some proteins that will bind to the free monomers to inhibit them from joining together to form actin filaments. Therefore, these proteins help regulate actin filament polymerization. The proteins will release the monomers making them free when more actin filaments are needed to be made. To assemble and make the new actin filaments, other proteins are needed. Actin bundling proteins are used to hold the actin filaments in parallel bundles together. There are actin binding proteins that hold the actin into a meshwork of the cell cortex. There are even proteins that will cut the actin filaments into small pieces. These small pieces are then converted into a gel fluid-like state. In this picture you see how the bundling protein and cross-linking protein are different. The bunching protein lays the actin out to form a sheet and the cross-link is at junctions. The actin is perpendicular to each other and binds them together to form a meshwork. In both of these the binding proteins are both in green to help them stand out from the actin that is reddish-brown. Lastly, we will discuss actin in the cell cortex. Actin is more abundant in the cell cortex than anywhere else in the cell. More specifically, it is most concentrated at the cytosolic face of the cell membrane. This is where the actin-binding proteins hold the actin filaments into a meshwork. This will strengthen the membrane and helps the cell be able to change its shape and move. In this picture everything that is red is actin. You can see in the cellular cortex, actin is the more abundant in the cell. I like this picture because you see all the similarities and differences between the three filaments. MTs and actin are more alike than IFs. They only have a few differences but not many. The next topic we will be discussing is cell crawling via actin. The first step is the cell pushes out protrusions at its leading edge. This step is driven by polymerization of new actin. There are two protrusions in this step, lamellipodia and filipodia. Lamellipodia are first in this step and they are a thin sheet that contains a dense meshwork of actin. The actin in this protrusion have their plus ends out towards the plasma membrane. Next comes the smaller and thinner of the two and that is filipodia. This protrusion contains approximately 10 to 20 actin filaments. Filipodia is not as dense as lamellipodia is. They both do have in common though that their plus ends are pointing outwards and they are both highly dynamic. We will discuss how they both are highly dynamic in step 2. At the top of this photo you can see the leading edge is zoomed in. You can see the filipodia is on the edge of the cell sticking outwards and helping guide it. The lamellipodia is directly behind the filipodia helping it lead the cell to where it has to go. Lamellipodia and filipodia both are highly dynamic because a cell that is crawling sends out structures to sense chemotractant and anchor onto a crawlable surface. The polymerization of actin into these structures that causes it to push the cell membrane out. When lamellipodia and filipodia thought the extracellular surface they intend to crawl on, they first have to stick to it. The transmembrane integrins help with them by adhering to molecules that are on the extracellular surface. Integrins will create an anchor of actin filaments on the intracellular side of the membrane. Basically, this means integrin will create a bridge-like structure of actin to the extracellular surface that is being crawled on. You can see the integrin is an orange and yellowish color. It is helping create that bridge-like structure to help the cell crawl. In the other picture the adherents are the little green dots there to help show that there are multiple to help the cell stick to the surface, not just one. The last step is how the cell pulls itself forward along the surface. For cell crawling to be promoted, a small G protein called Rho is needed to trigger the interaction between actin and myosin. Myosin is an actin-binding motor protein. Energy is needed for the myosin to move along the actin filaments, so they will bind to ADP and cleave it. Binding to ADP decreases myosin's affinity for actin. Myosin move from the negative end to the actin's positive end. There are two types of myosin. Myosin -2, found in muscles, and myosin I, found in all cells. We will be discussing myosin I here. Just like kinesin and dynein, myosin has a head domain and a tail. The head domain binds to the actin and cleaves ADP. The tail will determine what kind of cargo the myosin will be carrying. Myosin has two lever-like arms that are connected by a joint, much like a human arm is. When ADP is hydrolyzed, the joint will move. The lever will protrude straight out from the head domain. When ADP hydrolysis is done, it will be bound to ADP and the lever will be bent at a 90-degree angle. Here you can see where the cell was before it crawled and it is outlined in a gray dotted line. The next topic we are going to discuss are G-protein coupled receptors. G-protein coupled receptors carry a signal into the cell from modified proteins via phosphorylation. GPCR are the largest out of the family of cell surface receptor proteins. GPCR are sensitive to hormones, paracrine molecules, and neurotransmitters. Different signal molecules can bind to GPCRs. These can be proteins, small peptides, modified amino acids, and/or modified fatty acids. GPCR are commonly targeted by drugs. They are transmembrane, means they are found on the cell surface. They were synthesized at the rough ER and threaded through the ER membrane using start and stop transfer sequences. Then they are shipped and processed through the Golgi. From here, transport vesicle take it to the cell membrane. GPCRs are 7-pass transmembrane receptor protein superfamily. Other members in this family are rhodopsin, smell receptors, alpha factor receptors in yeast, etc. This picture shows an example of GPCRs being sensitive with hormones. You have the receptor that is labeled R and the hormone coming in to bind with it. When the receptor binds with the hormone, the G protein sees it and interacts with it now. Then a cell signal is released. When the GPCR changes shape, it activates the G protein that is associated with receptors at the cytosol face of the membrane. Even though there are several different G proteins, they all have in common that they bind to GTP and GDP. G proteins have 3 subunits: alpha, beta, and gamma. When the G protein is inactive, it is in alpha and binds to GDP. We know that when the G protein binds to the receptor, it changes shape, but then a secondary change in shape occurs to the G protein. The change in shape decreases its affinity for GDP and increases its affinity for GTP. When it's activated, it will let go of GDP and grab onto GTP. Once it is in the active state, the 3 subunits can target molecules. The G protein has GTPase, which means it can hydrolyze GTP to GDP. When it is done, the subunits will reassociation and translock it back to the receptor and repeat again. When activated, G proteins open membrane-bound enzymes or channels. For example, it will open ion channels. The 2 common enzymes that G proteins activate are adenylate cyclase and phospholipase C. In the next slide we will go into more depth on these 2. In this picture, the subunits are all this yellow weirdly circular shape. Alpha will bind to GTP and hydrolysis will occur, turning it into GDP. When this happens, alpha will interact with the beta and gamma subunits. Then when it lets go of the GDP and binds to a new GTP, the cycle starts all over again. Now onto a secondary messenger CAMP. Adenylate cyclase catalyzes simple reactions, ADP to CAMP. Phospholipase C catalyzes more complicated reactions. CAMP is a secondary messenger because it carries the message into the cell. CAMP only activates a single protein called protein kinase A. In the picture you see adenylate cyclase is embedded into a membrane. ADP is then turned into CAMP and it chooses to either activate protein kinase A or deactivate it. If it is activated, then a cell signal will be released. Calcium is another secondary messenger and will be the final one we discuss on this topic. Calcium uses another membrane component called PIP2. The signal molecule will bind to the GPCR. Along with the G protein be activated, it will activate phospholipase C. Phospholipase C will split PIP2 into 2 molecules, IP3 and DAG. IP3 leaves to the cell's interior. DAG activates protein kinase C. Protein kinase C phosphorylates the calcium channel. This allows calcium to enter the cell. IP3 will enter the ER and trigger the release of calcium from there. The calcium will bind to PKC, making the protein comedulin active. Comedulin will then activate another kinase. This picture has a lot of moving parts. Phospholipase C is a yellow oval shape in a membrane. When PIP2 is split into IP3 and DAG, they do go into different paths, but ultimately end up getting to the same place, which is cell signaling. Next we move on to the topic of CDKS and cyclins in the cell cycle. First, I am going to be talking about cyclins. Proteins are constantly turned on and off when they are needed and not needed. The cyclic regulation of proteins during the cell cycle is phosphorylation and dephosphorylation. Kinases and phosphatase activities are staggered. This means that some will be activated at the end of G1 phase while others will be at the end of the S or G2 phase. Kinases and phosphatases trigger the cell's profession through the cell cycle. Cyclins are a set of proteins that control kinases. Cyclin binds to the kinases and activate it, making it cyclin-dependent kinases. There are different cyclins that are made. When a particular cyclin is made, the corresponding CDK will be activated. The different cyclins are made in different phases of the cell cycle. Each CDK triggers the next cycle to occur. In the first picture to the left, you can see the different concentrations of cyclin in each of the phases in the cell cycle. In the next picture, you can see the cyclin bind to the CDK and the target protein to activate the next step in the cell cycle. Finally, we will talk about CDKS in the cell cycle, the way the cell stops itself at a checkpoint and from moving on to the next phase as CDK inhibitor proteins. This stops the CDKS from being activated and going into the next phase. DNA damage increases the expression and activity of a protein called P53. P53 is a transcription factor that activates CDK inhibitor, which is known as P21. P21 will bind to G1/S CDK to prevent it from going into S phase to allow for DNA repair to take place. If the damage cannot be fixed, then page 53 will cause the cell to undergo apoptosis. P53 is a tumor suppressor gene. If P53 is not available in the cell, then P21 can never be made as well. If there is number P21 in the cell, then the cell will not be able to know to stop to check for DNA damage before going into the S phase. This causes the damage to be replicated onto the new cells and it will keep replicating from then on. In the M phase, everything is distributed equally and then the cell divides into two. Segregation and cytokinesis require the help from microtubules and actin filaments. Everything that occurs in the M phase is controlled by a protein complex called MCDK. M-cyclin begins to be produced at the end of the S phase. It will then pair up with CDK. When there is enough M-cyclin, it triggers the formation of MCDK. MCDK activates CDC25 to be made, creating a positive feedback loop. For the chromosomes to be properly segregated, there has to be 2 copies of centrosomes. Each cell get a full centrosome along with the chromosomes. Centrosomes are copied in the beginning of the S phase. It is triggered by SCDK and G1/SCDK. Meiotic spindles form in prophase. MTs are unstable due to MCDK phosphorylating and the inactivate MT binding proteins. In this picture here, you get the whole big picture of cyclins and CDKS. You see the importance of CDK in each phase. It is needed to be able to get past that checkpoint to move on to the next phase in the cell cycle. The CDK is the brown bean shape at each phase. The cyclins are binded to each CDK in the phases and they are either blue, green, or red and are a crest shape. When they move on to the next phase, the cyclins let go and are degraded into little pieces. Our next topic are cell junctions. The first one we will be discussing are tight junctions. Tight junctions are cells tightly together so nothing can get through. This even includes water not being able to get into the junction. These are formed from proteins called claudins and occludins. These proteins bind together to form the junction of 2 cells together. Tight junctions are responsible for defining membrane domains as well. In this picture here, you see the proteins are in a string of beads and are a dark purple. They cover the enter part of the junction. There are spaces in between the two that are called intracellular spaces that look like air pockets almost. The next cell junctions we will be talking about are adherent junctions and desmosomes. Adherent junctions and desmosomes bind epithelial cells together. They both connect cells through cadherin. They tether to actin filaments or keratin, linking them to the cytoskeleton. Hemidesmosomes are a bit different because they bind epithelial cells to basal lamina. Together, all 3 of these create a sheet of epithelial cells that have mechanical strength. This is because they join the individual cytoskeletons together. In this picture you see a major difference already in the intracellular space than the one in the tight junction. This one is much more bigger. There also are no beaded proteins all over to help seal it up tightly. Here you have linker proteins to help almost zip them up together. The last cell junction we will be discussing are gap junctions. Gap junctions only exist between neighboring cells. They are a continuous channel between 2 cell membranes that is known as connexins. These gap junctions can close if they choose to because they are regulated like other channels are. This is where small inorganic and hydrophilic molecules can diffuse between cells connections. An example of a gap junction is a bead in a heart is created with the help of connected gap junctions. In the picture, again you notice the intracellular space is different than the adherent junctions. These one I would place in the middle of tight junctions and adherent junctions. This one also have channeled instead of proteins go help regulate it. On to the final topic of this e-report, and that is why cancers are selfish and do not follow the norm like other cells. Normal cells are social and live in large multicellular communities. Cancer cells do not follow this. They are asocial. Cells should follow these rules that are: cells should divide when a new cell is needed. A cell should live for as long as it can. A cell should maintain its cellular and biochemical character. A cell should stay where it is supposed. And a cell has no free will of its own. Cancer cells do not follow any of these rules. Cancer cells are you. They were your own cells that mutated and no longer care about you. They only care about their own survival. Cancer cells have two main properties to them. They are prolific and invade colonize areas that are meant for other cells. They can grow rapidly wherever they want to. Cancer cells do not require signals to thrive off of. They do not default to apoptosis. They are immortal, genetically unstable, have chromosomal mutations, are abnormally invasive, and they adhere, divide, and survive in foreign tissue. When normal cells are placed in a foreign area, they go into apoptosis, but cancer cells do not. In the left picture, you can see how invasive cancer cells are on our normal functional cells. They do not care about all the space they are taking up, and it is clear they will not stop for no one. They will go to great lengths and even make tumors and secondary tumors in a completely different area. In the next picture, you see how the cancer cells can have so many different distorted shapes. They do not provide any clues to their function except to live and fight our normal cells for dominance basically.